I'm on YouTube. Hey, welcome everybody to season 5, episode 1. What's potentially going to be an incredibly long season. The breeding project season. So, yeah, welcome everyone. Welcome panel. I know some people will be dropping in a little bit as well, but it's great to see everybody here once again. Made it through another week. Hey, alive and kicking. Always good, always good.
I'm not dead yet.
So on that quick note, before Lefty takes another hit and can't answer, Lefty, how you doing, sir?
I appreciate that, buddy. I'm doing good, man. Good week. Starting to trim up season 4, the peach pie and the Ethiopian banana X18 cross. I don't know if we updated last week. That ended up herming on me probably somewhere.
Yeah, you mentioned, yeah.
Hermaphrodite, I mean, probably somewhere week 3. Never saw it. Did a lot of hunting through the plant. Didn't see any nanners. Just must have been a random sack at the top of the plant somewhere that just popped and dropped. But yeah, it's pretty well seeded. Also, when I chopped it last week, I saw in the very top cola, there's a little bit of mold in it. So I have some adjustments to do in my tent. It was a new tent for this round. Actually, not even quite this full round, but had a very large cola, which is something I've never dealt with. So yeah, definitely something to think about in the future. Definitely need to get a little bit more air movement going, which I already got taken care of. But from the looks of it, it just stuck to the top to that top cola on the insides right there where it hugs that cola. But yeah, it's really sandy, gritty trichomes on it. So I'll go through, clean it up, and it's gonna get hashed, buddy. You know me, I like my hash, so it'll make a really nice hash plant. And yeah, looks like I got a pretty good yield of everything. Started trimming up today, and I'll be trimming through the show.
Nice.
Yeah. Sorry.
We'll come back to like the gritty trichrome thing in a minute, because that's actually quite interesting. It'll be interesting to talk about that. And what becomes interesting, I think, now about this season is stuff like gritty trichomes. These are, for the sake of what we're trying to achieve now, these become traits, things to take note of, because they may be indicators for other desirable things, or they may be what we're actually looking for. So on that note, swinging around, just before he's about to take a hit as well, Raptor, sir, how you doing? How are things going?
Hey, I'm doing good, everyone. Great to start off the new season. I'm very excited. Been wanting to delve into breeding ever since I first realized I could even grow this plant. So yeah, really excited. In terms of updates, 2 weeks into where I last cut off my plants and took cuttings, they're doing really good. I could have done a way better job at training. I just had my hands full with other things in terms of mycology. Yeah, my options for that are to raise up where I actually have a floating scrog. Shout out Purple Thumb OG for the idea. So I might even like lift it up and like remove, like not even remove, like, but just move around the things so I can establish that. But I need to take more cuttings going into that. Messed up all my cuttings. We got a little bit of a heat wave and got up to like 95 in my room. Everything died. Everything died immediately the day of. Yeah. So that's on me. People were telling me, oh, you don't need a special place. Yeah. No, it is what it is, man. I didn't have an established place to do all this stuff. And that's what I was telling people. I not only need an established place to do all of that, I also just need to work on my parameters of my cloning, my basic ABCs of cloning. I want to get that done, and I can move forward with the project with the scarlet begonias I'm really interested in. And I for sure got a couple males in there of scarlet begonias and Agent Orange. I only got one Agent Orange, and it seems to be a male. So no more throwing away males. It's testing them out now. Going through that. So I'll be setting up my tent soon. I already got the room that I'm going to be moving into to for my living quarters. It's all clean and set up now. I just got to move some stuff that I had for I was starting up other seeds and plants in that room. But yeah, that's all cleared, and now I just need to clear up this giant fucking bed that has enough, that's taking enough space for like 4 or 5 tents, 4 or 5 3x3 tents. So that's the whole thing. My talk to my family, they're totally cool with I don't know about the future blowing up of like all those tents, but I'm for sure gonna have another tent in there, and maybe even more as I look into my own mycology business. So gonna be making money off of that, but I need space and I need all the abilities to do everything. So yeah, that's what's up with me in terms of my mycology. Let's see. This is the pink oyster. It's doing pretty good. It's basically done. I'm going to be using this for my mushroom cultivation course, and I'm going to be taking the spawn and putting it on cardboard outside, and going to be growing mushrooms outside with this. So I'm taking a woodland mushroom cultivation course with Cornell. So that's pretty exciting. And yeah, that's about it for me. School's starting up. I'm starting to pick up pace with everything. No more getting lazy. I'm actually going to be planning on going on trips to the north on my own to collect mushroom tissue samples for my own
Nice.
library. So I'll be collecting spores and tissue and working on mushroom tissue culture. And in terms of tissue culture, I'm working on just reading through the basic books to get started on it myself. And I'm happy to share the knowledge.
Yeah,
This
I think
was all given to me. I'm more than happy to give it back.
that'll be
And
one that becomes particularly interesting later on. I think I mean, I know we've talked offline about it, but yeah, later on, how do you maintain stock of original
yeah,
genetics that you need to
once you
keep
got all
for the
this
whole
breeding,
breeding
once
projects?
you got all this breeding done, once you do all that work after 13 years of all that work, plus 13 years minimum
Of
of
everything
doing
you
all
had to
that
keep.
work, how do you save all that tissue? How do you save all of those things that got you there too? Because those things that got you there might be of use later on in your other breeding projects, and it might save you all that time. Why waste all that time? So yeah, looking forward to the future. This is going to be a heck of a year, heck of a season. Very excited. Thank you all for inviting me and including me.
Yeah, it's going to be a great one. And lurking in the background,
Lurky
quietly
lurk.
there,
Lurky lurk. How's it
GMF, how
going,
you doing,
folks?
sir?
Oh, can't complain, man. I'm above ground and I'm hanging out with y'all, so I'm great, man. That's the bar I set. Everything after that, meh. So yeah, I can't complain, man.
Excellent. How
I
did
get to hang with y'all.
perfect? How did that your experimental cob, not a cob, pan out in the end?
Well, I'll show you if you want. I got it ready for screen share. I took a macro so you guys could see. Everyone kept asking me what happens to the trikes. So I took a trichome macro so you guys could get a look at it.
Yeah, that'd be cool.
All right, screen share. There you go. That's what happens when that's an uncobbed cob. So that went in there. It went through the fermentation process. I can get into the nitty gritty of it if you want, but the long and short of it is, I gave it the standard time treatment. What I changed this time was the compression. When I did a rough mathematical calculation on the compression, it roughly has 2.6 grams of weed for every square inch of surface area. Whereas when you uncob it, when you leave it, just as is, and you try to ferment it, your surface area is something more like 0.025. So it's like literally 12 times more surface area on an uncobbed cob. And so that's speeding up what I think is the rate of reaction. And as you can see, the trichomes amber off kind of very uniformly. There's no variants. That's not, some are milky, some are clear, some are amber. They're just amber, and they're all one color amber. So I'm pretty sure between the way it's being packed is affecting the rate of fermentation. I think, and this is just a rough calculation, this is the equivalent to like 3 or 4 year old weed now.
Nice.
Because it wasn't pressed, right? That pressing is really what the best way I can try to explain it is by compressing it, you buy time. When you compress it down to that cob and then you bury it, yes, it takes a year to ferment before it's ready, but that means you've got another number of years where it's going to stay almost at a very slow rate of progression, right? Until it becomes this. If you don't press it, the rate of reaction basically takes place over the entire surface area. It doesn't have to work its way through the density, and the actual rate of reaction is drastically sped up. So yeah, like I said, my guess is this is, it's roughly a factor of 12 that.
And have you tried it yet? Sampled it?
We're just about to, actually. I saved it for you guys. So I'll show you here. This is, let me give you a live shot of what the non-macro looks like.
And there's an 8-minute warning apparently, so that's probably in the how do we?
So if you can see that on the plate, yeah, there you go. That's what it looks like, not under a macro. So it looks like brown stuff you wouldn't want to typically smoke. And there was a spot I noticed on one of them where it lit there. I think you can see it on the back there, that little white spot. It had just started to sporulate a tiny bit of powdery mildew, but then it stopped as soon as I dried it. So I think that's part of letting it go so long too. If I'm going to do this again, it's going to be much, much shorter, like by a factor of 12.
Okay.
And then next to it, you can see these leaves, right? These are also something I put through fermentation with a little citric acid. And now they don't crumble, they're not brittle, and they're like cloth. You can fold them, you can scrunch them up, but then it'll fold back out to its original shape, no problem. So I think this is how we're going to be able to make these into rolling papers, is by giving them a similar ferment, so their texture becomes something pliable, but it's actually much tougher than when it started. It won't rip, it's not nearly as brittle. So that also went for, these fermented for roughly a week. So, and during this one's week fermentation, I would probably cut this down in half, maybe even in third. Like I said, I did the math and it's about a factor of 12. So I may even literally cut it from 7 days down to, 14, 15 hours in secondary ferment if it's uncobbed. And that should be all it takes. Or it may be a color indication thing. So yeah, we've got some more experimenting to do. But as you guys saw on the macro, it definitely, it's doing something. And it's doing something very uniform. And it's getting us a very uniform trichome profile. And if I don't squish it, those heads stay in nice shape. They're nice and round and they, glisten. So yeah.
So that probably makes it interesting for the extraction guys at that point when it's fermented in the hole rather than squished. In terms of making it convenient for the home grower to be able to actually mass prep weed without the smell and all of that, and the issue with high humidity to deal with it, then cobbing it is probably, pressed cobbing it is the way to go, because you can get so much into a jar, and it doesn't create the odors, etc. But yeah, that looks really interesting, and that's one that I think we will definitely have to take a look into it further. So kind of getting into the swing and the topic of season 5, which is going to be all around the plant breeding side of things. And I figure would probably be a good way to kick it all off is like just to, let's go over some of what, like the real basic side of it, getting some of the terminology down. Again, this is going to be one that runs for quite some time. So there's plenty of room to have quite a few discussions on it and get there and cover a few other aspects of it.
Get yourself a notebook, get yourself a glass of water, settle in.
Welcome. There will be a test at the end. Da-da. Hey, almost semi-professional. Reminds me of those old style books. So, I mean, yeah, certainly back, it used to be a case of, you cross the best for the best, etc., and you hope that you get the best out of it. And is that actually always the case, and are there better ways of doing it? And definitely over time, we've provided, we've certainly developed better techniques when it comes to plant breeding in terms of how to do selection processes, what we're looking for, how quickly we can identify things. So on that side, it's definitely moved forward quite a bit. Just for the case of the bottom here, what this does, this has created, of course, when you look at it today, where you've got genetic modification, where you can do splicing, you can mix and match things, you start to split the styles of plant breeding into what would be known as conventional and unconventional categories of plant breeding. And the unconventional being doing it in a lab, where you probably need a lot more equipment. It's not something you can necessarily do at home. Not right now, anyway. Who's to say in like 5 years' time it probably becomes a lot different. But for the purposes, I think, of what we're doing in this project, it's really going to be looking at the conventional side of plant breeding, which is what I think we're all capable of doing. And it's the one everybody can do with almost anything that they like. And moving on from that, I guess we get into the big one, is like genotype versus phenotype, and what are the differences between the two. And so genotype is an organism's full hereditary information. So that's every single gene combination that you have in your DNA, essentially. That's your all of the information that's stored in there. Phenotype is your expression of that genotype, as it's stated here, as an organism's actual observed properties such as morphology, development, or behavior. What is important to understand about phenotype is it is the genotype plus the environment. So the same genotype can produce a range of different phenotypes simply by making alterations to the environment. This could be purpling expressed when you drop the temperature, which it would not normally express if it stayed all temperate and warm. Fox-tailing under high temperature stress is a phenotypic expression. Not everything will necessarily do it. Some will, some won't. Some will even do it regardless of whether or not it's warm. It's something that they will just do. At any point, if anybody has any questions in chat, whatever, please fire away. So when we're doing plant breeding, everybody wants to cross things. And there are two types of crosses that you can get. There's what they call divergent crosses, and there are convergent crosses, which we'll get to in a second, which come after this one. And the divergent crosses is probably the one that most people are familiar with, in terms of A crossed with B gives you AB. I'm crossing these two plants together to give myself a single offspring. Not offspring, yeah, offspring. And you'll see in a single cross, you're crossing these two parents, like two traits. This is really simplified at a very high level, but the proportion of the genetics from each one is 50-50. So the offspring has 50% of the genetics from one side, 50% of the genetics from the other side. And then when you get into a three-way cross, so it's A crossed with B, the progeny of that was crossed with C, resulting in a mixed one that's got A, B, and C in it. And because of the order in which the crossing was done, you see it's only got the final prodigy has only got 25% of A and B, but 50% of the final one that it was crossed with, because that brings half the genetics to the table each time. And then the double cross, as you can see, when it gets down to the final one, it's quarter representation from the genetics for each of the original parents that we used in creating that final plant. convergent crosses. This is the back cross. Okay, there are others that come with it, but they get really complex. And I think the one that most people are familiar again would be something like the term back cross, and seeing it in, just so everybody's aware, it's BC, not BX. Okay. And here it's a case of you cross the first two parents, that produces your first filial line, your F1 of the AB cross. You take that first cross and you cross with parent B in this case. The generation from that, you cross that with parent B, and you're always crossing it back with the same parent, original parent. And what you actually see is over time, the proportion of the plant that is that original parent on that first back cross, it's 50-50. And suddenly it becomes 75%. And the ratio starts to ramp up really, really quickly until you've almost got back to the original parent again. You have a very small bit of the genetic pool that's not parent A. And there are so back crossing is used for very specific reasons. And the divergent crossing that we looked at a moment ago, that's also used for specific reasons. You've got different end goals in mind while you're doing those two different processes. And we'll get into that and discuss that as well. And then just quickly on getting into some common notation. And I think this is probably the biggest one, the F symbol and its usage. All that the F actually denotes is the progeny of a cross between two parents. So every single seed is actually the F1 of the two parents every single time. You know, that's in a very generalized term, that's exactly how it would be used. What becomes important is when you're trying to breed is you're actually using that as your inbreeding lines. So F1, I take parent A, parent B, cross them together, I get an F1 generation. I take plants within that F1 generation and cross them, I get F2s. Cross those within those F2 generation, cross them, I will get F3s. The second you step out and bring in a plant from outside, you're back to the first generation of that line being worked. So you have your inbreeding crossing that you're doing, and each time you do that, the way to remember it is the seed is one generation ahead of the parents. So F2 parents produce F3 seed. F3 parents produce F4 seed, et cetera, et cetera. And so it keeps on going down that. And selfing F2s produce F3 plants, et cetera. So long as it's really just a way of tracking where you are and making sure that, because what you'll discover and later on we get into it, it becomes a thing about numbers, it becomes about ratios. And if you start to mess these things up the wrong way, you really throw that out. It becomes very difficult. You'll eventually get down to, oh, hang on. And that's where the back crossing comes in again. So yeah, it's going to get interesting. And then the symbol for back cross is BC. And it's a progeny between an original parent and an F generation from within that line. So yeah, this will get built on, and we can get into talking about like Punnett squares and that as well, which become another interesting way of being able to track traits and doing things with traits. You have single traits and multiple traits that you're trying to track. You can get into some of the downsides of trying to use Punnett squares, for example, when it gets really complicated. But yeah, just on that, I mean, any questions, thoughts, ideas? What do people? Does that change some people's ideas of like how this actually all strings together? Is it like, oh, hang on, I didn't think of it in that way?
I really like it, especially the usage and teaching everyone, not just us in the group, but also the people following the Punnett squares. And like what you learn with that 4 Punnett square, that's just for you to basically understand what the way of going about it, but it can get into huge variables, and that's what it takes. I remember even asking in class about it, uh, for my biology classes, and they're just like, "Oh, you don't have to worry about it. That's for breeders." And I wish I would have pressed and said, "No, I'm looking into breeding." I should have just said, whatever, flowers. I want to look at petunias. I want to look into breeding that. But whatever the case, I would learn the basics. It's all the same thing. You all have to learn the same basics in order to progress properly. But yeah, I really like it. It seems that we're on the right path, and especially since we're using materials like literature to go back on and reference, just like how your background right there, Pete, like that explains everything that's going to be happening, and it's just one page. That's not even one page, it's half a page.
Yeah.
So the in-depth, I'm glad that we're beginning to become more in-depth with it, and not just like referring to it like as like some ghost in the wind, like, oh yeah, reading, yes, the F1, I hear it in the air. uh so yeah, I'm very excited. uh what do you think, Black Sail? By the way, how's it going?
Hey, Keith.
Hey, how's it going, guys?
Good, good. Yourself, buddy?
Oh man, busy. You
It
know,
looks like you have one of your lights there. Keith is always well lit.
I just have the blinds open. But yeah, for some reason my face is really glowing right now. But Pete, I see you got a bunch of
He's
charts
pregnant.
and symbols up there behind you. That looks fucking cool. Yeah, really good to be hanging out, y'all. I think I'm just about done for the weekend. About time to start drinking and fucking around for a day and a half, and then back to the grind. So very happy to be here.
Nice. Yeah, that's always good. Always good to have you here, sir.
Uh,
I see
we do
Richard
it-
was asking about S1s. Now, actually, it's no
Exactly.
different to an F1.
exactly.
An S1 is just the first filling. It's the same. It's same. Now, what I will go into next week, actually, there is a subtype of notation that can specifically be used in some of these situations. I'll actually produce that for next week. I'll create another slideshow for that. And if anybody is interested actually in like where a lot of this information is coming from, I can highly recommend the Principles of Plant Breeding, of Plant Genetics and Breeding by, I can't pronounce this guy's surname. Hang on, can I get an angle that'll work? Fuck it, let me turn off the virtual background. So I can highly really recommend that. As it's not about cannabis, it is about plant genetics and breeding. So it's not something that has to be unique to cannabis. In fact, these guys have been doing it commercially in that for a very, very long time. And I think what becomes interesting as well is to see how much they have to go through to bring a new plant to market. It's not a quick process. Sorry, GMF.
Well, I was going to ask, why don't we talk about that a little bit, about other plants and bringing them to market? I think you exactly nailed it, because a lot of us don't know. So let's just, take on the assumption we see how this chart works, right? How would this chart apply to, say, a specific trait you want in a pepper, or something like that? Like, how would walk us through how this chart would apply to something we're looking to do in something other than cannabis?
Okay, well, now we're going to okay, so the next thing to get into is understanding then of I suppose genetics in terms of dominant traits, recessive traits, and this actually steps into the Punnett square side of things. So actually, on that, I'll put this link up in chat. So this is a really good site. Great the Punnett square calculator. So what you're going to have to decide on is, are there traits that I'm trying to track through a plant? And if we were going to use a what kind of trait would you be looking to do in a pepper? We can come up with some labeling here. Let's say we want it to be hot.
Tall.
Okay, and we got another one. So let's say the peppers could be hot, or they could be hot or mild.
Can we put an expression of morphology or something like that in there, or color? Could we say like we want them hot and purple?
Yeah, okay, but that's two traits you're looking for. Let's just start off with
You just want to stick with one. Okay.
And let's say what we want plants that are hot. Now, what you have in genetics is you're going to end up with dominant genes and recessive genes, where the dominant gene expresses itself over the recessive gene. And usually what you'll find is if you've just genes appear in pairs, especially what we're looking at here in this plant, genes are in pairs, so you'll have this combination. If you have a dominant gene and a recessive gene in the plant, the dominant gene expresses itself. So you don't know that the recessive trait is there. You wouldn't see it. It's not going to express itself. The only time a recessive trait, and again, we're simplifying it here, the only time a recessive trait appears is if both parts of the gene are made up with the recessive trait. The chromosome carries both of the recessive genes. Otherwise, you won't get it expressed. The typical way of doing the nomenclature for it is to use a capitals for the dominant side, and lowercase represents usually the recessive trait. So we're going on about having like genotype and phenotype. What's interesting is in this case, let's say we took a pepper that's hot. So we know it's a pure breeding hot pepper. So it's a dominant pepper, and we have another pepper that's hot, but we don't know its genetics specifically. When we cross the two, you wind up with two different genotypes that come out of it. So if we only got 4 plants, we've got 2 genotypes that came out. Still 2 pure bred hot plants, and these 2 sort of hybrids where they've got the recessive and the dominant gene hiding in there. Well, they've got the dominant gene with the recessive gene hiding in there. So it's 2 different genotypes, but in terms of the phenotypic expression, there's only one phenotype being expressed. So when you grow the plant out, they all look the same. You can't tell the difference. The only time you'll suddenly spot the difference is when you take that generation. So these are the F1s that got produced from this cross that happened here. This is your F1 generation. If I happen to pick the hybrid, well, the one that's carrying the recessive trait, so I pick those 2 for the next cross, I will suddenly get 1 plant out of the 4, and it's just like ratio-wise, we're really basing it here, but 1 plant out of the 4 will suddenly appear not hot at all. I'll have these 3 plants that are hot and 1 plant that isn't. And that would tell you, from a breeding perspective, hang on, one of the parents had this recessive trait. If you never see the recessive trait appearing, then you know nobody's got it. But this is the importance of the F1 cross produces cross the F1 generation to produce the F2 progeny. That F2 progeny usually has the biggest variation in the group because all of the recessive traits can suddenly start to appear. Breeding projects usually start with the selection made out of the F2 generation. Examples being if you're looking for recessive traits, you won't find them necessarily in the F1 generation. They will only start to crop up in the F2 generation. Freak Show is an example of that. The one with the like fern-type leaves, that is a recessive trait. Anything you cross it with, the F1 generation from that cross does not express that heavy variegation of the leaf, but go into the F2 generation and suddenly you see it appearing all over the place again. Now, this was just one trait that we were looking at. You bump it up to 2 traits. It's just automatically filling some in here. but you'll notice straight away it gets a little bit more complicated, and that's now just 2 traits. And so I want it to have strawberry smell. I want it to be short flowering, but I want it to be really resinous. And you can see how complicated this gets. It really starts to, and let's say I want it to go purple. We're up to now 54 different phenotypes being created from just having 4 traits that we were looking for, which generally gives us how many genotypes are we going to play with here. So a 4 trait cross creates 81 unique genotypes. And yeah, and it just gets the number gets sillier and sillier as you bump it up.
And I think this is actually raises a salient point from E in the chat, and this is where marker-assisted breeding comes in, because it lets you sort through all those variables much, much faster rather than having to go through the time and effort of growing them all out and seeing what the hell they are.
Exactly. This is where the modern techniques of being able to do DNA fingerprinting, whereas you could now just take that sample, DNA fingerprint it, and go, oh, okay, yeah, that has the traits I'm looking for. I'm going to keep those plants instead of having to physically grow them the whole way out to see those traits. And what's vitally important, again, is that you're growing all of these things constantly in the same condition, because remember, phenotype is genotype plus environment. So if you start messing the environment around, suddenly you're going to get a different phenotypic expression from that same genotype, which maybe makes you not selected all of a sudden because it didn't look quite the same, for instance. So that bit becomes equally important all of a sudden, is keeping environment really locked in as well.
So in case somebody's curious, I broke that up, and it does look like brickweed even when you break it up. It's dark brown. So.
And the smell of it?
Smells really fruity. Really like super ripe fruit. That's again, this strain kind of smells like fruit anyway. It smells like kiwis, kind of like gooseberries. It smells much fruitier now. It just smells, yeah, like really ripe fruit. And break it all up. But yeah, so it just looks dark and like brickweed you probably would turn your nose up at. So we'll have to wait and see. I'll tell you in a minute.
No, excellent.
Yeah,
and then I know I was mentioning the length of time it takes to do this. Again, it's not an overnight process. It's not like you suddenly bring out a new, at least let's say you're in the commercial grain market or producing a new pea or a new strawberry or something like that. You want to bring a new strawberry to market commercially, it's got to be better than, it has to bring something to the market that's better than any other strawberry that's commercially on the market already. You can't just bring just another strawberry. It's got to be yield better, be more pest resistant, mold resistant. It has to have something that's improving what's already there. So the kind of trials that those guys have to put their stuff through is really intense. And at the same time, trying to, this one is what to say, like generalized steps for breeding a recessive trait by backcross methods. So trying to lock in a recessive trait can take up to 11 years to do. Now we go back to the great debate on should a seed breeder get financial compensation when you use their intellectual property to start your seed empire, because you just shaved 11 years of R&D of getting your breeding empire off the ground. And it can take longer than 11 years to bring the stuff and stabilize it. I think Freak Show, the guy's been working on that for almost 2 decades now. Yeah, that's a lot of hard graft that goes into it. And I think that's when people start to appreciate when you bring, a new strain to market. a truly new cultivar to market. A lot of work went into it. So yeah.
Yeah, that's crazy, man. I'm like, my default position would be that it's a lot harder to like do the work and maintain the company and market and, you know, make a reputation for yourself and all that. And that, you know, someone starting off with your seeds, like, I try not to sweat it. And I think it's kind of silly when people in the grow light game sweat it, like, oh, someone copied my design. I'm like, man, you know, it's a pretty fucking simple design anyway. But dude, like 11 years of work, man. Can you imagine just on your first release, somebody just snaps it up and is like, let me just crossbreed with this fucking gem.
Yeah. You're gonna be round.
Yeah. Oh,
And now, you know, like earlier we were looking at like, okay, so, you know, like a single cross, like A and B, to create, if you were working the two A lines, the A line and the B line, so that when I cross A and B, I get the same thing every single time. It could have taken you 20 years in total, because you've almost taken 10 years to do A, 11 years to do B, and then you bring them together. You know?
okay.
Yeah.
Well, you had 11 years to get your name out there and to get like a loyal following. And when you drop the Fire Genetics, people are gonna fucking, you know, they're gonna copy you. But I guess the only thing you can do really is like position yourself to be the name brand, you know?
Yeah, yeah.
Because what else are they gonna do? Like, are they gonna, like, if somebody puts a seed out on the market, is somebody else gonna be able to get a genetic test on that and be like, hey, you know, I've got these copyrighted genetics?
You can actually do that.
Absolutely.
And interestingly enough, it's okay. So the funny thing is, this is, you can't patent what occurs in nature. You can patent what does not occur in nature. And when you work a new line and create a new line, that is considered does not occur in nature, which is because you've had to do like really heavy selective breeding to get to that endpoint. Sure, people will argue, you know, it's the same thing, enough monkeys and enough keyboards and enough time and you get Shakespeare's work. But, you know, in the general scheme of things, okay, yeah, maybe in 30,000 years time, that might appear naturally in the wild, but it's just not naturally occurring. And so what you patent then is the bit that does not occur naturally. They do that with apples and they defend it quite successfully.
Monsanto, they do it with corn. You know, and here in the U.S., if their pollen gets in your field, they actually now can come after you for that next generation of seed, because that's their genetic property.
But there they do it with genetic markers to be able to track it.
Mm-hmm.
Yeah, they mark it that way.
Yeah, but that whole if their corn pollen end up in your field, it's theirs kind of thing. It's kind of bullshit, especially if they're doing any kind of outside or outdoor pollination. You know what I'm saying?
Oh yeah, it's terrible. It's terrible.
Like, what the fuck?
Well, but I mean, look,
fight.
it's happening in cannabis though. Look at it. Like, you know, I'm sure some of us have had, you know, a plant outdoors that ended up seeded, and you're like, oh, mystery pollen.
Yeah, that happened to me actually. Yeah, with the cross I was working on last season. Yeah, at the end of fucking last season, a male popped up. So yeah, in theory, on paper, there should not have been any males because it was a reverse female pollen onto another autoflower female, and boom, there goes a male. It was done outside, so I guess that just proves that there definitely is straight pollen looking around.
Yeah.
So yeah, lesson learned. Won't do that outside again.
And of course, if you're trying to do a breeding project, that really throws a spanner in the works, because you suddenly pick that outlier plant thinking it's a genetic outlier of of these parents that you've crossed, and you've suddenly found, you know, the unicorn in amongst everything, to quote the idea of You suddenly find it, but actually it's not the parents. And no matter what you try and do, you just can't reproduce it.
Yeah. And then like, well, at least for me, I was lucky enough that it was just the first cross, so I could have afforded to just scratch that whole fucking project one time. But I mean, if I was balls deep into that, that would have been a different story.
Now I see like Bill Sid in chat saying that that's why they're selling F1s. But yeah, every seed you buy is an F1 in theory, by the nature. What's different is if you ask the breeder, is this a dihybrid F1 or is this an inbreeding line F1? Those are two very different F1s. And that's, for instance, what the difference is between convergence and divergence breeding. Convergence with the use of the back cross is actually what you're trying to do is you've seen a plant that you've got this plant, it's got 90% of everything you like in it, and there's this other plant that's got just this other one trait that you want. And you're just trying to add that trait to your plant, and then you will have like the 10. Let's say you've got an 8 out of 10 plant. The extra bit that needs to make it a 10 out of 10 plant exists in plant B. So you cross A and B together. It's going to kind of break the lettering that was used a moment ago. You get the first generation of that, but what you're trying to do is you're still trying to have everything from the plant, your plant, that, the 8 out of 10 plant, you still want everything from that. So that's why you now back cross back to that plant, and you do it again and again and again. And each time you do it back to that same original plant, you then increase its percentage of genetics in the line until eventually you'll do the cross one too many times, and that extra bit that you were trying to bring in disappears. And you've got to go back one generation and go, okay, that's as far as I can actually work that particular trait. And it's now everything in that generation will start to express it. The other one is where you bring two lines together to create something completely new each time. And that is your dihybrid F1, where you would breed like 50% essentially from the plant A and 50% from plant B.
You mean 50% height by 50% height? Sorry. Get you chem dog. Sorry.
And that's usually where you see the hybrid vigor kick in. It wouldn't be in doing the back cross piece to create this new stable line. That real hybrid vigor would kick in the dihybrid F1 that gets created. And the thing about that is it's always the same. So you may actually use two back crossed lines stabilized to create the two parents that you then cross to give you that dihybrid F1. So again, it's just how many generations are you trying to do this for? And then it comes down to a numbers game as well. You can't do this with, so you can't. It's really difficult to do it with 5 or 10 seeds at a time. You look at big commercial guys, they're doing 10,000, 20,000, 50,000 at a time. So they can see the variation and work out the ratios. At some plant breeding is a statistics game. That's really what you're trying to do. It's a game of numbers and ratios. Yeah.
Well, and I think go ahead, Raptor.
Oh, I was just gonna, when I was hearing you guys talk, you and Duckweed talk about breeding outside, that just brought up a whole nother can of worms. Like, if you really want it to be adapted to the outside, do you do the breeding outside as well? Obviously, you do the selecting out, but yeah, it's just something that came across my mind. Like, but would it be of a benefit at all? So yeah, it's something that came up, and I was just waiting till the lull came in to bring it up because I was actually interested in what both of you were saying. You were saying something else, Jima?
No, I think that was funny enough. I was going to actually get to that. I'm kind of on the same mind as you. I think that's part of exactly knowing we're now playing that game between, are we experiencing dilution of genetics by being outdoors and having random pollen land in? Because even one of my students this year, you know, it literally is the first year people can grow, and he had seeds. He has no nanners anywhere. You know, he inspected it all over himself, but even he out in near the woods ended up with 1 or 2 seeds, and he had no nanners anywhere. So even just this first year that people can do it, they're throwing them out left, right, and center, and they're obviously not paying very close attention. And it's lighter than air until it bonds with a water molecule. You know, that stuff will just off it goes.
Mm-hmm. I mean, look how it's all over the world. I mean, get there. It got spread around quite easily.
Well, and that's really its genetic drive too, between being able to do these amazing things like, you know, root in weird places, root in different soils, you know, pass through animal tracks and then sprout, down to being able to pick up pollen from literally miles and miles and miles away. This plant really wants to survive. It's developed even to being able to go intersex when it needs to as a survival mechanism, rather than necessarily as a trait. That is in itself just, you know, the kind of genetic drive this plant has for survival.
And all of these, you know, actually people taking like the cookies and then taking platinum tangy cookie and crossing it with something else, like crossing it with Gorilla Glue, you know, Malibu Pie, you know, headband skull fucker. All you're doing, those are just diluting the gene pool. That is not strengthening the gene pool in any way. That is actually what is making it weaker and weaker and weaker. It's not introducing genuinely new stuff into the mix.
But that's what everybody is doing. There's almost every breeder is doing that.
If you take one hybrid and cross it with another hybrid and cross it with another hybrid, unfortunately, it's not a hybrid vigor each time. You're actually cutting it out each time as you go further and further down. You need to bring in, like, even the royal family occasionally has to bring in something new. So yeah, that is the kicker. But I hear people, I mean, I've seen it in chat, people have said, you know, is that part maybe of why we're seeing strains aren't quite as good all of a sudden? You know, people long for some of like the really good ones from yesteryear that don't seem to be around anymore. You know, like if you want plants as tiny little nugs, okay, yeah, maybe they smell great and everything, but is that all that you're looking for? You know, is it worth it? I mean, I've seen people talk about growing some of the latest hype strains and then going, it's just not worth it. The yield isn't there. You know, great, it smells nice, but you get an 8 that smells nice, and it took me, you know, 12 weeks to grow it. Yay. You know, is that really what we're after? Anyone? Bueller?
Well, I just think the takeaway for me from all this is that I will never ever be a breeder, because this shit is really complicated, and I would just end up probably chucking pollen, getting whatever I get.
Well, and that's what most people do. I think that's just the long and short of it. Most people don't take the time to even sit and, you know, look at their plants. I take a lot of photos of my plants, but I don't really post them just because I take photos to look and kind of, I'm curious. I want to study it. So I think it really comes down to the mindset, though. Most people just want weed. Yeah,
and the plant count, right? I mean, you need a really large sample size to do this properly. Probably, I would have to get some kind of license or some shit. I don't know.
Or you have to hit the genetic lottery. That happens too. That's kind of like people. I'm amazed every day at the amount of people that buy lottery tickets. Right? This is essentially what you're doing. Exactly possible.
Yeah,
you're just playing odds.
We all grew Orangegasm though, and I'm wondering if any of you guys, there's this phenotype that I didn't save. It was too late by the time I realized it was my favorite. It was like a medium size for Orangegasm, but the buds were a little bit darker. They had a little more purple in them, and it's got just the fucking most incredible, like sweet, funky orange flavor. Like that first hit of a fresh bowl is like out of this world, man. I don't know if any of you guys saw anything like that.
Nope.
Not from the Mystery Grower side. I know there was some talk about ones that smelled like Sunny Delight, was it? That kind of creamy, sort of orangey sort of smell.
Like orange creamy soda. Like I had one that smelled like Trix cereal milk. That was
Nice, man.
my favorite out of all of them. Was like the creamier orange smell.
The nugs are different too. I'll go grab one. I'll show you.
The Mystic Grows in chat, and I've been watching on her Instagram feed recently. She was collecting and storing all the pollen, which is something that we've discussed. You know, what's the successful way of doing that? Which again, that's a nice way of being able to not have to keep the, you know, if you can store pollen successfully for 10 years, 11 years plus, suddenly you don't need to keep that male around. If you were able to keep, you know, 10 little vials full of pollen, that's enough pollen to see you through for a very long time. And again, from a plant count perspective, that could help out. And this is where Raptor, when we get into the tissue culture stuff, again, how do you, you know, maintain the plant count? Oh, let me spotlight that.
I don't know how well you guys can see it, but the nugs are darker.
Mm-hmm.
They're really dense and they're darker and they're a little more purple.
Nice.
It's just a little bit different from all the other plants. And I wish I had saved it.
It's always the case, isn't it?
I've got like a quarter pound of it though, man. I'm gonna smoke the shit out of it while I can because it makes me super happy.
Good,
That,
good.
That orangeism has some really gritty, like dry resin heads on it too.
Yeah, definitely.
You break up a bowl, and I mean, it leaves a pile of keef behind anytime, you know, you put it in your grinder or break it up in a tray. I mean, you pack a bowl and it just leaves a huge pile.
And was it Husky in chat? Sorry, just looking through some of the questions there. Yeah, I mean, that's the thing about land races. Generally speaking, you know, bringing a land race into the mix is good. It brings in all of this original material again that hasn't been diluted down. What have you got there, Lefty? Which one is that?
This is a super crop of that peach pie.
Definitely can see how they all take up. And that's what part of the purpose of super cropping is, to get all of those buds to flip up.
And to keep it out of your light.
Very nice, very nice.
Good stuff.
Great training.
Yeah. So I know the Mystery Growers got the cross-pollination. Well, the pollination has been successful and already seeing seeds forming on the plants, which is going to be interesting. Yeah, I know they're looking to try and reduce the flowering time of the line that they want to work, and that probably turn into a back cross line in terms of just trying to lock in that trait and then keep everything that comes from the K4LV2, which I think is the tracking name that they use for it. Yeah, it should be interesting to see. How's everybody else getting on with them?
I have a dismal report. Those older seeds I tried to germinate, zip zilch 0 on the first 5. So I went through and I watched Pat from Green Bicycles. I watched his video on germinating old seeds, and now I'm having to start to delve deep into all the different methods of scarification and where and why, and you know what different combinations of stuff people are putting on them and weird because you know now it's time to. I was just hoping they'd sprout in dirt like everything else does, but no, it is really old stuff. The stuff I'm working with, this old, brickweed bag seed, it's going to be kind of a crapshoot. But I already did decide on a name for it when it's finally ready. Like when I finally make a cross with this Mexican bag seed, it's going to be my Platinum Punch, Cookie Cake Crunch, Gorilla Skunk, Banana Runts, Jack OG Skittles, AK Diesel Chunk. Huh?
Sounds about
People
right.
are gonna love it.
Sounds about right.
They're gonna love it. I don't even know what it's from because it's bag seed, but they're gonna love it.
Just hit us with the acronyms. We'll be fine.
I got some PPCCCGSBRJOSADC. That's hype enough for you? I'm trying to be hype.
Yeah. So well, seeing that what I was working on for the last season was fucked. I got some what is it, Slurricane crossed a GMO Mac cross, and then back to the Slurricane. So it's a BC. And then, but those were the F2s, by the way, back to the Slurricane. And then on the other side, I have some TKM 10s crossed with Platinum Pies. So I'll sift through those, and yeah, I just pop a couple in each for the new year. And yeah, so far everyone is looking healthy and nice. Some of the Slurricane Mac GMO cross, some of them are looking a little bit iffy in terms of, they have kind of funky leaves. So probably I'll give them until the end of January and see what they do, and then pop some more if there's anything, if I need to.
Yeah. And that's, you talk about the leaves, that's been an interesting discussion that's been going around as well, is even like talking to Jeff, he says he spots the phenotype that he's looking for quite often by looking at the leaf structure. That gives him a really good indication of what he can expect to see in the plant. So there are going to be certain things where, I know we've talked very basically about dominance and recessive traits, but what you have is not quite as simple as that. And like somebody in chat pointed out, like blue eyes is a little bit more complicated than just a gene. It is there is more to it, especially if you get into green eyes and things like that. It gets even more complicated. So we do have the concept of like co-dominance, and where some traits are linked, they're not co-dominant traits, but they're linked traits. So you can't get one without the other. And that's often those kind of cues that we're trying to look for in the phenotypic expression. How easily could you look at it? Oh, this leaf structure means it will have good resistance to powdery mildew, for instance. Now there's no way of doing that other than by doing the test. So you suddenly discover, everything that has a really serrated edge is good. As an example, has good powdery mildew resistance. With this big spoon-shaped leaf, suddenly that, if you discover anything with a big spoon-shaped leaf is going to be 30% plus THC, as an example. So those are these linked traits that come together where you can't, every plant has leaves. It's one of those things. So yeah. I see Mystic Grows tips there for storing pollen, using silica packs to remove the humidity. And I think what's oxygen remover to remove the oxygen as well, which is going to be great. I wonder if you could replace it with nitrogen gas, but then of course you'd have to keep doing that every time you opened it up. You'd have to replace it with
Well, something we do in brewing is because CO2 is heavier than air, you can purge a vessel and very quickly use it. As long as you don't agitate it too much, you can say I'm going to fill a growler for someone, I'll typically purge it with CO2 first, then run the beer in, because the CO2 is heavier. It'll form its own blanket underneath the air and displace the air. So you could definitely do that with CO2. It's really easy to do. You just need a CO2 tank and little.
I see there Tasmanian devils managed to store pollen for 2 years without the beads. So that's good. I mean, that's good.
Really Ozzy wanted me to give it a shot, so I took some, and I just thought to myself, okay, if I was looking to store it, what would I do? And just totally out of an experiment, I mixed the rice with a little bit of cornstarch, because that's one way I know the cornstarch will deal with any moisture before it gets in there, and that'll actually help stretch it out a little bit when I go to use it, because I didn't harvest that much. So I just mixed it with a tiny bit of cornstarch before I sealed it, and then I put rice on the outside of it. So,
yeah, and the good tip as well from Mystic Rice, if you do when you're storing in the fridge or the freezer when you take it out, is let it warm up to room temperature before you open it, so you don't get any condensation suddenly occurring due to the pollen being cooler than the air temperature and being below dew point.
Mm-hmm.
Yeah, but
ruin
well,
all
I mean,
of
considering my situation, if I take that out to the freezer and leave that on my kitchen counter, in 2 minutes that whole thing is gonna be fucking full of condensation.
Not if you leave it closed, though. Now, if if it's still closed, let it warm up to room temperature. But for you, given that your ambient temperature, ambient humidity is 90%.
Yeah, that's just it. Because I mean, like I've tried with other stuff where I would put stuff into the freezer in a couple of bags, and no matter what I do, if I leave it to try, there's no way I came across where I could take it out of the fridge and get it down to room temperature without having basically condensation inside the innermost bag, and everything in there, all the contents in there fucked.
You would probably almost want to
I don't know.
open it keep it in the fridge, which will be fairly dry, and then open it in the fridge where it's still cold in the fridge.
Yeah,
dip an earbud
in. I might,
earbud's worth of it, and then sprinkle it out really
just
quickly.
Man up and open it in the freezer and keep your hands in the freezer.
I mean, well, I do have a 6 foot long deep freeze. I could probably get in there with it.
There you go. I know you're not used to frostbite, but you'll be all right. Like you said, once it gets to room temp, you'll warm right back up.
Yeah. So yeah, because of that now, that's why I if I do try to cross anything, I try to have everything to line up with each other, so I could collect the pollen here and then reuse it on a different plant right here that's already ready to get that.
That's right next to it. Mystic saying a sealed room with a dehumidifier running at probably full chatter, and do it in that space.
Oh yeah,
doesn't it make more sense then to just keep cloning your male though, so you don't have to worry about it? And you always got a little dude or willing who's mature enough because he's cloned, so he's more than 60 days from seed. He's always willing to throw for you.
Yeah, who was it that was saying that struggles with keeping males going, or cloning males and keeping males running for a very long time? Is it as easy as doing cloning females and keeping them going for that long?
Good question.
I mean, I think Jeff's been running that Solomon male for many, many years now, actually. Is it? Yeah, it must be the same cut still going. I guess when we get deep into the tissue culture stuff, that's where you can revigorate the plant by going down to the tissue culture route and starting up again, because that's a true new fresh clone. Create at that point.
Sometimes they get male that would just flow out on you in 18.6 under 18.6. So you do those,
Well, what I think is going to be really interesting, I've been hearing a lot of things from more like Russ the Bob, for example, was remarking on how just putting the plant outside, giving it natural sun, if you can put it in its natural setting for whatever duration, one season, how it brings back and how it fixes a lot of things. If you have some issues, if you were to just give it some sunlight for possibly a week, you might see a lot of those issues dissipate.
He said run it outside for a season.
Oh, a season. That's
I
what
thought he said not. I thought he said run it outside for, and that makes sense, because you're bringing you're bringing up its photosynthetic potential back to where nature kind of intended it.
Yeah, I've heard that exactly like what you're saying for a season in terms of fixing it just in general. But I have heard from people that are growers that they just put out their plant for like a couple of days to a week.
Oh, maybe he did say that. Maybe I just misheard him.
Probably. I've heard this from Dude Grows show mentioned. They just like said, "Oh, I just put it out for a week and it was fine. It just fucking fixed itself." So there's something to say for that sunlight alone. I'm sure Black Sail Market can agree. Like that's a great, like that's the goal. We can just get to that sun, like recreating the sun in the tents. That'd be amazing.
But the
honestly,
heat to get rid of.
Yeah, I got some heat to get rid of. A little bit.
Honestly, I don't know.
UVC is going to be rough.
I don't know if that's if copying the spectrum of the sun is actually ideal for the plants. Like I think the ideal spectrum is defined by the McCree curve, with that bump in blue, and then it goes down to like 60% in green, and then a bump up to like 100% in red, and then back down. The sun is more of like closer to almost like the measurement for lumens, where it's just a big old spike in green and yellow and a lot less on like the red and blue side. So I don't know, man. I guess the plants just didn't evolve to like, they have different pigments, and each pigment responds to a certain wavelength of light, like blue or red or green or whatever. And those pigments evolved as a result of random mutations. So chlorophyll has a big spike in its absorbance in blue and in red. It doesn't match exactly the spectrum of the sun.
There are 40 different types of chlorophyll. So chlorophyll evolved in plants. There are 40 different ones that respond to different wavelengths of light. It's not the same in everything. So
in cannabis, though.
Yeah, but you hit the nail on it because, for instance, dark plants. So it's photosynthesis in cannabis and other plants is the same. I mean, it's no different in that term of the chemical process. But the ones that put out black leaves, they're actually tuning into a different frequency than the ones that put out green leaves. So it just in cannabis, it's like that. What's it, the Black Rosa, I think it's called, that the whole plant is actually kind of almost dark purple black to the point of it's almost black. That's something that's trying to probably absorb a lot more warmth. And maybe it's after a different spectrum of light that it will respond to, simply because it's actually the reason why you're seeing the purple is it's absorbing everything. It's absorbing all the green light. It's not reflecting any green. The reason why you see green is because it's the spectrum not being absorbed. That's why you get to see it.
Well, it's not that it's not being absorbed. It's just being absorbed about like 15 to 25% less than the red and the blue. So but the plants still very much utilize green light.
Oh yeah. I mean, you don't see
anyway.
but the reason you see green is because it's being reflected. Everything else is being absorbed through. So when you see purple, the green isn't being reflected anymore.
Right.
And photosynthesis goes back to before plants, like way, way before the first. I think that's why they call it a cyan, like the common ancestor to cyanobacter or whatever it was. That was the thing that evolved photosynthesis. And when plants evolved, they got it from that bacteria, just like we got mitochondria from bacteria in our evolutionary chain.
Yeah, you'll get plants that have no green in them per se that photosynthesize. It's a really interesting. Green light's a high energy particle as well. It passes clean through the leaves quite comfortably. So that's why they would. At one point it was like, oh, green light, and that's why you can use green light. It doesn't affect plant growth, etc. Maybe it doesn't affect it from a photosynthesis point of perspective, but it does absorb some of that green light energy in terms of it's passing through the plant. It gets picked up as it goes through. And it's like these things. It's like the entourage effect. Not having it, you can't put your finger on, or when it's there, you can't put your finger on why the plants are responding better. So when it's not there, it's like a really odd. An interesting one of those would be, just as an example, Gollum from the Lord of the Rings. Not Lord of the Rings. Yeah, Lord of the Rings movie. Sorry, I'm thinking Hobbit. So Gollum from Lord of the Rings, when they did some initial tests with them, they didn't put on, they're called subsurface scattering. So your skin, when light hits it, scatters the light underneath the surface of the skin. That's what makes it, look the way it does. And they would turn it on and off and ask people what was which one looked better and why did it look better. And they would always pick the one that had the subsurface scattering enabled, and they could not tell you why. And it's just like one of those subtle things. It's there. It has an effect, but you can't put your finger on it unless you know exactly what you're looking for.
It's like fish sauce or, in food. If it needs to be in the dish and it's not in there, you'll know it. It won't taste right, but it's nothing that you won't be able to pick out. That's why it's not right. Or anchovies too. Like if you don't have anchovies in your Caesar dressing, it's not right. It won't taste right. But you won't know that's why it's not right.
Remove the fermented pilchards from your Worcestershire sauce and it won't taste like Worcestershire sauce.
Exactly.
Most people go, sorry, what now? What's in here? Fermented what? So yeah.
Like fermented sardines as well, right? Something that
I
think I was watching the how it's made on that. I was just like looking at all. It just had a full list of, I don't even know, over 20 for sure. Things in there. Just, oh my gosh. Yeah, that's a great way of looking at spectrum too.
Well, and,
yeah, Gma.
well, no, I was just saying, keep in mind that took a long time to make too. The original Worcestershire sauce was a failure, and it was actually something that Lee and Perrin abandoned. And it was many years later they found it in the basement and said, "Oh, screw it, we'll taste it." And suddenly they were like, "Wow, this is gold." So this is also something that takes time as a ferment to actually generate all the pathways necessary. And they didn't necessarily know what we do about microbiology. They can't turn the knobs like we can turn the knobs when we ferment beer and all these other, like last night I made sauerkraut. We know how to turn the knobs. They didn't know how to turn the knobs. But now that we do, that's the real key to making some of these things amazing, is taking them through these biological and microbially managed processes. That's how miso gets the miso. It's how the umami gets the umami.
How was the sauerkraut, man?
So far it's okay. It's just started. It's basically, I took some of my, this is funny. I went and took, 360 grams of shredded cabbage, which is about half a cabbage, goes into a quart jar. 10 grams of pink Himalayan salt, because I'm going for about 2.5% salt by weight. Then I did 45, I did 45. I did 70 grams of a kombucha, my kombucha starter. So I just went into my mother, went down to the bottom with a sanitized turkey baster, pulled out 70, went into each jar, and then the rest is water. And that'll start the ferment. So it's just a quick inoculation, but yeah, it's a cheat. It's already bubbling. So, like I said, I like to cook with the cheat codes on. Same thing when I brew. I like to brew with the cheat codes on. It's a lot more fun.
Oh, brilliant, man. Well, I must say, I'm really looking forward to this. I think it's going to be really interesting and exciting. And I hope people in chat also get into it and follow along. And I know, like, for instance, Keith, you made a comment earlier about it seems really complicated, but I actually think there's a lot to be said for, there's nothing wrong with throwing shit together and seeing what happens. Quite often that sort of like sparks the curiosity, like, wow, look what I actually got out of it. And if you think, how much do people charge for a pack of seeds? I mean, I know I'm not, and I admit to it, I knock the idea of, just flippantly crossing stuff, but only when it comes to releasing it commercially and putting that shit out in the wild. That's where it becomes an issue, because you're just confusing the gene pool. You're not making it easy any easier for other people. But at home, if you're just fucking around, I mean, you could save thousands of dollars, pounds, or whatever by doing a little sideline breeding project of your own. You know, one plant produces hundreds of seeds. And who's to say you don't get lucky, you don't hit the lottery? And you know, a lot of these amazing strains that are out there were people who just happened to hit the lottery. Pure flukes. I mean, that's how like almost 9 times out of 10, the best thing just happened by accident. You know.
Well, and let's talk about the real benefit, though. The real benefit isn't the lottery ticket, because yes, you're right. You could get the lottery ticket and win the $1 billion strain, but the real benefit, is if you pay attention, you can learn stuff. And it's hard to always, you can have something explained to you theoretically, but when you can do it with your own hands and you can make the process, you can watch it, you can take it in, that lets you learn in a way that is not always accessible. So that's another reason I really think it's worthwhile, because if nothing else, it's kind of like when I teach people to brew beer. Even if you don't, brew all the beer you drink from now on, you appreciate a lot more what someone else does to make you a good beer. Or if you cook at home and you really like to cook, then you really appreciate a chef who's really good at their job and really puts their heart and soul into it, because you know what they went through to do it. So I think that's the same thing.
And on this show for this season, it doesn't have to be cannabis. I mean, if somebody is like, I really would like to try this out with chilies, go wild. It doesn't have to be the same. I mean, oddly enough, the same principles apply. The big difference on that is that the chili is self-pollinating, which does introduce some interest. But a lot of those plants, you will discover you can still successfully cross-pollinate them with a different strain, species, or cultivar, because the actual different parts of the flower will mature at slightly different stages. So you often find like the female part of this plant will mature first, maybe the plant next to it. They'll kind of all do it slightly out of sync. So although they can self-pollinate, they sometimes don't, just by offsetting the sequence in which parts of the flower mature up at slightly different stages. But yeah, I mean, that's exactly the same as it is here in terms of that makes no difference. And I think that's a great way of doing it. Oh, that's odd. Not letting Duck back in, is it? Oh, yeah. He'll have to get home. How bizarre.
Yeah, very odd. He said he's restarting the phone, so we'll see if that was the issue.
So yeah,
longest food trip in history.
Exactly.
Right.
Oh, there's Mystic Rose say Jack Herrera made about 7,500 seeds, and the other plants made around 4,000 seeds each. So yeah, you'll be busy for a while if you're doing this at home. It's definitely a really nice part of it, and you can share them with some of your friends, I guess. What would be really cool to see at the end of this whole series or this season would be everybody able to produce a stable line out of it and go, that's it, and anybody can actually see the full story behind how those lines were created.
Oh, there's nothing to say we can't do more than one too. I was kind of thinking about that the other day. Like, if you really want to run one or two side by side, if you've got the same parent, you're gonna like the same father you're gonna use or something, that you can keep 2 alive next to it, go for it.
Oh, hell yeah, yeah. And look, part of this is going to be people are going to get someone go, shit, I've got to start again. That is just the nature of all of these things. It's an experiment. It's not. Failure is always an option, as they said on MythBusters. Do you need to shoot, Keith? Yeah.
Yeah, sorry guys. I do still have some shit to do today. So I got to take off in a minute here, but
have a good day, bud.
Yeah, and I mean, you might be able to talk me into it. I know that at first glance this stuff can be overwhelming, but I think you and GMF are right that if you just start doing it, it will all become clear a lot more quick than you think. You know,
telling you, dude, you can do it.
You're already doing it. It's just a matter of finding a good male and keeping a good male and figuring out what it is you want to pair it to. I mean, you're already doing it. You're already growing, Keith. I mean, you're already going through it.
Cool, man. Yeah, maybe one day.
We'll all be standing at Indo Expo the entire. It's Jeff going, "Hey, these fuckers all started with my shit. What's going on here?" The Irie Army Road.
He already said he was cool with it. He's said it many times. You guys are welcome to use it, he said.
I checked with him.
Yeah.
That's why I invest in Irie.
He's really good, dude.
Man, it's always good hanging out. So thanks for inviting me. Have a good weekend.
Absolute pleasure.
Go
See you, buddy.
You, Keith. Stay safe. Catch you next week, buddy.
Later, Keith.
And actually, on that note, we almost brought it to an hour and a half. But yeah, if anybody would like to, we're going to kick it off. We'll drop off the live stuff. We'll carry the after show going on here for a little bit longer. So if anybody in chat would like to jump over and join in, here is, I'll post the link up once again. You are more than welcome to dive in, join us, join in on the chat. As I said, it goes offline now. So yeah, this is all good. Some closing from everybody. Lefty, you bastard. He's trimming away. Bastard.
Lefty's Farm on IG and Cannabis. And I'll give you an update next week on my breeding project as it starts to grow again.
Fantastic. Raptor, sir, where can everybody find you?
Raptor Grow, all one word, all lowercase, Instagram, and you can find me Tuesdays, Wednesdays with Humidor and the Flavors, 7 p.m. Pacific Standard Time, and tonight, actually every Saturday, 8 p.m. Pacific Standard Time, Brews with Buds. Lots of fun. We'll see if I make it. My new schedule, yeah, coffee is going to be a big factor in making it. So I might see you, Pete. If I don't, it was awesome having you. I'm sure. Actually, I'm sure I'll see you during the week later on today. You know. So yeah. Awesome chat. Lots of great questions coming in throughout the show. Thank you so much for. I'm trying to become more active in chat. It's a little hard focusing on both, so I'll do what I can to help Pete as well. Look for questions that are directed, more towards everything. So yeah, looking forward to the entire new season. It's going to be a good one. We're going to learn lots and help each other. That's the goal.
Yeah. Judging by the looks of the diagrams we look at earlier, it's going to be an 11-year-long season, boys. Buckle up. Buckle up.
Get comfortable with each other.
Jesus, 11 years from now, I'm just thinking, fuck her, hell.
Still on season 5.
Yeah, a bit more grey, I think, by then. Probably a lot less up here.
Probably all of us, and the Gandalf beard going.
They might even have legalized it globally by then.
Right.
Yeah. We'd be able to send
and we
stuff to
can
each other.
expand it onto multiple projects.
Maybe there will be no such thing as a plant count anymore.
Oh God, don't tease me. Get me all Randy.
Whoa. We all have 4 plants that are legal.
Exactly. Yeah. There's a plant count around chilies? What the fuck?
Yeah, right.
Speaking of tomatoes.
How about you, Duck? Where do people find you?
You can find me at your mama's. And
awesome.
Yeah.
Tell her I said hi.
They'll be there.
I give her a big fat hug. Yeah. Just everyone in the chat and on the panel. It was nice. I don't know what anybody said before me because Zoom was being a shit on me just now. So, so yeah, it'll be a pretty long one ahead. looking forward to it though. Looking forward to learning a lot more new information, on the whole breeding aspect and all that jazz goodness. And thanks to everyone in chat for popping in and hanging out. Yeah, you can find me at nutinguts868 on Instagram. If you figured it out, lucky you. If you can't, well, sorry.
And GMF, so where can everybody find you?
Typically, if you got a question or something I can be helpful with, find me over at Instagram, Genetic Memory Farms. If you guys are curious, I don't know, let me know. I'll post about I took pictures at least, and I guess I should post it, on how to make a quick sauerkraut at home, just with stuff you had at home. I used a mason jar and a plastic bag. That's it. And a little sandwich bag. So it's stupid easy. You could even use the dregs from a kombucha you drank and pour it in there and kickstart that fermentation, or the end of a bottle of live vinegar. So if people are interested, I'll go ahead and post that on my IG. Why not? So we'll make some sauerkraut together. Yeah, just hit me over there if you got any questions or anything I can help with, any brewing questions, food questions, whatever.
Excellent. And you can find me at Peel Grow Systems on Instagram or at Operation Grow 2019. Yeah, and I'd like to say a big thank you to everybody in chat who was helping out on the questions and that this evening. So Tasmanian Devil, Mystic Grower, thank you so much, and along with everybody else. I'm just picking Magma Seeds, thank you. Richard D, thanks for the book that actually provided a lot of this information. That's where this bits and pieces are coming up from behind me here. And if anybody has anything in particular that they would like to potentially cover or delve into, ping a message over, let us know, and we can get involved in that and see what we can do. If anybody either in the panel or in chat maybe knows somebody that, a good breeder, again, it doesn't have to be with cannabis, it can be with anything. I really want to get some of the big commercial, I say big commercial, like commercial established plant breeders and the normal commercial industry to come and talk about it, but how they do it, what their processes are. I think we can learn a lot from them in the structured approach that they have to take. It would be really interesting to hear some of the challenges that they face in bringing something new to market, which clearly is not currently an issue here, but maybe there's the writing on the wall for when this does all go perfectly legal. These are the things people will have to adhere to. So yeah, on that note, if anybody,
is on the wall. The writing is on the wall, people. Have faith.
And if anybody would like to jump in on the after party, please feel free to follow the link and dive on in. So on that note, thanks everybody. We'll catch you all next week once again. Stay safe and blaze it up.
Yes.
See everybody